So, well, my name is Cosma Brian. I'm from Maseno University. And today I would like to bring you this topic, erythropoiesis. Erythropoiesis is the normal production of red blood cells. And it normally takes place in the red bone marrow. So, it normally takes place as a result of hypoxia. And let me write the word hypoxia. Hypoxia. Hypoxia is a condition whereby you find there's low oxygen. What makes the oxygen to be low in the blood can be as a result of anemia, excessive bleeding or blood loss, or higher altitude. Maybe when you are climbing Mount Kenya or Mount Everest, there's low oxygen levels right there, guys. So today we are going to see how erythropoiesis, the genesis of erythropoiesis. First of all, let us come to the GIT. Yeah. So let's first just highlight what normally important for erythropoiesis to take place from the GI. We have folic acid, we have B12, we have fats, we have amino acids, we have the carbs, that's carbohydrates. And we have iron also, very, very important, iron. So, let's start with iron. When iron comes into the stomach, it travels into the, let's say, this is the duodenum, around here, duodenum. Then let's zoom in into this duodenum. Zooming in into the duodenum to see what normally takes place here, the duodenum. At the duodenum here, there is an enzyme here, known as reductase, ferroreductase enzyme. Let me write here. Ferroreductase enzyme, known as duodenum cytochrome B. This duodenum cytochrome B converts duodenum cytochrome B converts this iron 3 positive to converts, after iron has come through this process here, it converts iron 3 positive to iron 2 positive. By the help of hydroxy ion, it enters through this channel. How do we call this channel? This channel is called divalent metal transporter 1. Metal transporter 1, yeah. Divalent metal transporter 1. Inside here, there is a protein here. This protein is called apoferritin. This iron 2 positive binds with this apoferritin to form ferritin. This ferritin is I2 positive. This ferritin leads to formation of a polymer here that is known as hemosiderin. This is iron 2 positive, hemosiderin. So this iron, this ferritin can also be moved out of the cell. This cell is known as enterocyte of the duodenum, enterocyte. So moves out. After moving out, there is a protein here. This protein is known as hephaestin. Hephaestin. This hephaestin normally converts this iron 2 positive to ion 3 positive. This ion binds into transferrin, a protein here. This protein is in a sinusoidal capillary that is found in the bone marrow. This is a capillary here. Now, this ion goes into the transferrin, I think that is the transferrin, somehow, transferrin. This transferrin is just a protein here. This ion comes in and binds in there. So we have taken in ion. Now let's take in B12, vitamin B12. Let's take in vitamin B12. I have a big task here because I'm recording at the same time I'm teaching, so I have a big task here. So taking in vitamin B12. In the stomach here there is parietal cells. These parietal cells have an intrinsic factor. This intrinsic factor binds with B12. After binding with B12, it comes all the way to the ilam. It comes all the way, I'm sorry I didn't show you where. It comes all the way to the ilam. Let's say the ilam is located here, and let's zoom in to the ilam. At the ilam here, because the B12 was bound into the intrinsic factor, now it comes all the way to this place here. Let's say that it was here, that intrinsic factor, and now let us say that this is the let's say that this is the intrinsic factor. The red one is the intrinsic factor. The intrinsic factor is the red one, and the blue one is the B12. Now, through a process known as receptor-mediated endocytosis, now this comes in. The B12 now comes in. B12 now comes in through that process of receptor-mediated endocytosis, and then it's taken all the way to the capillary where it binds to transcobalamin 1 and 2. Now inside here we have ion 3 positive. We have B12. So also other things like the amino acids, carbohydrates comes in, and then they are taken in. Now inside here we have the amino acids, we have the carbs, carbohydrates, we have fat, we have iron inside there. So I think now you have seen how it normally comes to introduce the GIT. So there also, let's come into the liver. Also the liver also has a channel here, a transporter here. This transporter is transferring, same to this one. Now this transferring, normally ion comes and binds here. Ion comes and binds here. After binding, the hepatocytes, remember the liver has an hepatocyte cells. The hepatocyte cells produce hepcidin. This is very important. This regulates the activity of this channel. Oh, I forgot to tell you about this. This channel is known as ferroportin. This channel is known as ferroportin. So it regulates the activity of ferroportin. So without this, this one, when ion is in excess, this one stops it normally blocks ferroportin from function. It normally stops it. That means that this one regulates the activity, regulates the amount of iron in the blood. Remember, iron, when it accumulates to certain levels beyond the required standards, it's normally toxic. So there is also a protein around here. This protein is known as HFE that normally regulates the activity of hepcidin. Now I think we have all the requirements for erythropoiesis now in the capillary. Now let's come to the kidney. The kidney is very important in production of something we call EPO. Now let's zoom into the proximal convoluted tubule. Let's zoom in. So in the proximal convoluted tubule, there is EPO, there is a gene. There is a gene. There is a gene here. And also there is hypoxia inducible factor. Let me draw it like this. This hypoxia inducible factor, hypoxia inducible factor, it's normally bound into an enzyme. Let me draw it like this. This is an enzyme that is bound to this hypoxia inducible factor. It's normally hydroxylated. So that tells you that oxygen is very crucial, very important. Oxygen is very important. When oxygen is very low, or when there's no oxygen, this enzyme normally releases this hypoxia inducible factor and to bound. It normally releases it to bound with this gene here. After binding with this one, now EPO is released. EPO, erythropoietin, is now released into the bone marrow. Now, in the bone marrow here, we have hematocytoblast stem cell. Hematocytoblast stem cell. Hematocytoblast stem cell. This one can differentiate into myeloid and also lymphoid stem cell. So this myeloid can differentiate into three types of cells. We have platelets, we have WBC, white blood cells, and RBCs. Platelets. Now, because EPO normally commits this myeloid stem cell to form red blood cells, the specific one, red blood cells. Now, this one triggers the formation of red blood cells. Normally, the first cell to be formed is pro-erythroblast. It's pro-erythroblast. After this one, to these two, because there is something that we call the DNA transcription to RNA and RNA translation to proteins. Translation to transcription and then translation. So this one, after this one, we have the formation of another cell. I'll tell you what its name. It's normally called basophilic. Basophilic. It's normally bluish. Basophil, basic. This is due to transcription of DNA to RNA base. This bluish, because of RNA. From this one, we have another cell. As you monitor this cell, you see that the nucleus is just reducing in size. I'll tell you the reason why. So this one is It's called this is called basophilic erythroblast. Erythroblast. This one is called polychromatic. Polychromatic. The reason why is that some of the RNA has been translated and some of the DNA is now being transcription of DNA is going on and translation of RNA is going on. That's the reason why polychromatic, because it is red, there is red, and also there is bluish. Some of the RNA not yet translated. So that's the reason why it's called polychromatic erythroblast. This is basophilic erythroblast, polychromatic erythroblast. Now this one forms the one with now red and very small here nucleus. It is very red and small percentage of RNA. Now this one is called autochromatic erythroblast. Now this one, remember, it has a nucleus, but this nucleus and all, it will be ejected out. Let me draw it around here for you to see. Now this is the autochromatic erythroblast. Here's the nucleus. This nucleus will be ejected out. And also the organelle, the mitochondria. The reason why is that this one takes in oxygen, pyruvate, and takes in oxygen. That process of aerobic respiration leads to the production of ATP. That means that if red blood cells were to contain this mitochondria, that means that this oxygen would have been used, and we don't want oxygen to be used. We want this cell to load oxygen to tissues. And also, now when it has been ejected out now, these cells now that are just ejected out, the nucleus and also the organelle, like the mitochondria, now it comes and enters into sinusoidal capillary. Here, they develop into reticulocytes. These are reticulocytes. Reticulocytes. Now after that, they develop into red blood cell. It's complete. Now the reason for B12 and fatty acid, amino acid, carbohydrates, and iron. B12 is essential for maturation of DNA and the DNA synthesis. DNA maturation and not synthesis, but DNA maturation and condensation, all of those, this is function of the DNA. Even amino acid, carbs, and iron is essential for heme synthesis. Now, I think this is the entire process of erythropoiesis. And I think now you have got the overview of it. We started with diet, we came to iron, and comes in the duodenum. We zoomed in the enterocyte. We said here there are reductase enzyme, ferroreductase enzyme, that normally converts iron 3 positive to iron 2 positive. Under the help of hydroxyl ions, it gets into the enterocyte through these transporters, divalent metal transporter 1. After getting in, it binds to apoferritin. After binding to apoferritin, there is the formation of ferritin. Ferritin forms a polymer here, that's hemosiderin. Ferritin also can get out of the enterocyte through a ferroportin. This is a transporter here. The getting out, there is also a protein here, epohistine. Epohistine converts iron 2 positive to iron 3 positive. It gets into sinusoidal capillary. After getting in, it binds to a protein here. So after getting in, others also get through the GIT, goes through there. B12 comes in, binds to intrinsic factor. After binding to an intrinsic factor, it comes in through a receptor-mediated endocytosis, comes in here. Then here we came and discussed here about this kidney, proximal convoluted tubule there is, and there is a gene here, EPO gene. Also we see the factor here, hypoxia-inducible factor here, and now this effect, now it comes in, assimilate the stem cells to start the production of erythropoiesis, entire process here. So thank you for watching. Welcome. I'll be producing more videos, making more videos for us to understand this cause. Thank you so much. My name is Cosmo Brian.